The last 30 minutes here before we send you on your way was to have a brief panel recap. So I guess Klaus and David, if you can hang out here for a second. Who else were we? Nakissa, Eric, would you like to join? And
well, because I'm a creature of habit, I will go ahead and mention everyone here in the room, please do use the microphones if you have questions. And for the virtual attendees, please do use the Q&A feature in the room. And so, we wanted to come back and close out today and reflect on what we've kind of learned over the past day and a half around what we see as reasonable and available contexts of use using models today, as well as what level of support and what level of evidence is needed to move those forward. And so as we kind of move into this, reflecting a little bit, I would like to start off asking each of the panelists up here, what your thoughts are at this point after a day and a half of discussions around this, and quite a bit of discussion with, I think, every talk touched on a context of use. Why don't we start with Klaus?
So what aspect is the most important piece to get to a framework, right? Because that's the ultimate objective. We want to have a framework that acts as a compass for the ecosystem to be able to properly frame context of use statements for what could be transformative complex in vitro models. So what are the most important key aspects of that? What we heard throughout the 2 days is understand the question, but the question needs to be driven by a tangible reality of drug development the answer to which needs to add value, and it needs to inform a meaningful, tractable decision that needs to be made in drug development. You start with that, and then you can follow the framework that Tina put together and that she alluded to in her talk, and that many others presented components for throughout the 2 days. What is the baseline information you have to make that decision today? And why is there a gap in the certainty? How can a complex in vitro model fill that uncertainty? By how much? It doesn't need to fill the entire underlying uncertainty. It needs to fill the uncertainty to a certain point where you can say, I'm going to be comfortable with this decision. That starts from a very tangible understanding of the drug development needs from industry. Then it needs to factor in with the regulatory requirements. And then that provides the spec for the developers to be able to generate those solutions in that meaningful way. If we get to a point using the liver as the example, I think we can expand this to whatever toxicity is relevant in drug development. That would be my take home there, and I'll leave it at that.
Okay, thanks. And Nakissa?
So I think what I got from these last couple of days is that there's a greater sense that we are passing sort of this initial period of hype about these models, and we are trying to find how we're going to apply them in a setting to help the development of drugs. I think that they've been used to some extent in discovery, but they really haven't contributed to the development of drugs from the perspective of the FDA's involvement, because it's been for those types of data that are typically not received at the FDA. we are looking for ways to see how to use this sort of new technology, which incorporates sort of like a three-dimensional construct of cells, to get additional data that we didn't have before. And so I also think that there is an understanding, it's a greater understanding now, that animal models are not going to be replaced anytime soon, which I think that, you know, maybe a little while back there was a lot of hype about the fact that these models were ready to replace animal models. And I think that there might be more of an understanding that that's not, you know, where we're at right now. We are trying to complement the data that we get currently, both from a non-clinical as well as from a clinical perspective. And these tools are being seen as adjuncts to what we are currently using. And the goal is not, you know, the 3 Rs is good. We want to, everybody wants to get to the 3 Rs. That's not the goal, though. The goal is to develop better drugs and safer drugs and drugs that work better. And so how are we going to use this technology to try and get there? If we can manage to meet the 3 Rs in the process, that's a good thing. But I think the priority is to really, you know, get better drugs. So I think that shifting the focus away from replacing animals to seeing what are some of the endpoints that we cannot currently address, you know, with the methods that we have, is one thing that I'm getting away from this meeting, that there is general consensus in how we're going to use these tools for that. I think the other thing is that I think being able to all agree on the fact that using the example of DILI, that immune-mediated DILI is something that cannot be currently measured. It's a complicated process, obviously, that is not, we're not going to be able to address it immediately, and maybe for many years. But I think that there are gaps that can be filled immediately with the models that are currently available, if some modifications are made and some you know, standardization is made to the approaches that are being used. And I think that they are things that can be worked on. And so there is a likelihood that we might be able to get to a point where additional methods for assessing the mechanism of drug-induced liver injury via the immune system could become part of the battery of tests that are available in trying to rule out what are potential causes of something that is seen late in development. So from my perspective, the discussion here sort of brought us a little bit closer to the reality of the field, as well as to a closer endpoint for adding to the types of data that is included in the kind of like when the DILI group that Skip mentioned, the consult review that they have, they look at a bunch of methods that they evaluate. So there's, from a concrete perspective, there is an additional test that could be added in there that could be recommended to sponsors, and I think that that would be a good addition to you know, what exists right now. So
yeah, thanks, Nakissa. Yeah, definitely. I think, you know, where there's unmet needs, and when a potential issue arises, you know, it's specific cases. It's not always every case every time. Eric?
I began the workshop, pardon me, hopeful that animal usage could be minimized, you know, these 3 Rs. And then at some point, it occurred to me, well, even if we get these DILI models perfect, 100% of everything, well, there's still all the other organs that you have to think about with drug development, where animals are probably still necessary. And that brought my expectations back to reality. A main take-home message, I think, from what I learned here, is that the conversation has to start with the modelers. The modelers have to make us understand what's possible and what's not possible. And from an industry and development perspective, hopefully I impressed upon you yesterday that the unmet need spans the entire spectrum of development. There are unmet needs everywhere. And whatever we're able to come up with here, industry will find a use for it somewhere within the whole process of drug development.
Thanks, Eric. David?
Well, I think there's been some very good comments here so far, and really enjoyed the workshop. My takeaway is figure out how to be useful. I mean, if you want people to adopt a new tool, solve a problem for them. And that's really, I mean, this question of interest is like, solve a problem for me. So the drug development tools qualification, and you have to understand the customer if you're going to solve a problem for someone. So like drug development tools qualification, who's the customer? It's sort of a review team at FDA. And then, but it's not just FDA, then it's the pharmaceutical companies have to find value in it too. We consider that at FDA when we're considering qualification submissions. It's what's the unmet need. And I'm not just talking about just complex in vitro models. If you're developing a tool for certain class of drugs, you know, do we expect there's going to be a bunch of drugs developed in this area? You know, we can only do so many things at FDA and like reviewing qualification submissions. So we want to, what's the return on investment? And are you solving a problem that's going to help move the needle somewhere or make somebody's job easier, bring drugs to market faster? There could be lots of problems you're trying to solve, but like, yeah, figure out how you can be useful is sort of my takeaway.
Thanks. And, you know, kind of back to Eric's comment, not around all the, well, actually I have a comment there too. For anyone who was at the MPS World Summit and saw Donna Mendrick's keynote address, I think she opened up, you know, talking about how many different tissues are tested for histopathology in tox studies. But as well, the other piece, now I'm having having a moment. No, and oh, this is what it was. So, you know, going back to around asking what the developers can do, and a conversation I had yesterday with a few people is you know, I think that one of the actions we have coming out of here is looking at is putting together at first a simple Venn diagram of what is available and possible right now today, and what we see as a prioritized list of unmet needs, and then see where we have something in the middle that we can begin to advance. You know, we went through some of what we thought were today and identified where there are additional complexities within those, and ways that we can approach that. But I think that an exercise like that will be very useful.
That sounds interesting, and I would add a component to that of those things that are available, having a marker for high uncertainty or low uncertainty, right? Because when the uncertainty is really high, more people are risk averse. So even if it's available, if the uncertainty is high, people aren't going to buy into it. So that would be a good piece. That sounds really interesting.
That's a great addition. Thanks, Tina.
Yeah, and I'll just add sort of the way you can, how do you become useful? You have to like talk to the other stakeholders, and you know, so hopefully that was one of the goals of this meeting, and hopefully we can have other avenues to facilitate interaction among different stakeholders, and because you kind of have to be useful to a lot of stakeholders to get buy-in across the board. So it's something to think about.
Thanks. Well, I'd like to open it up if there's any questions or comments that people in the audience would like to bring forward right now. And I'll warn you, I can do a long pause. Not quite that long. All right. Graham? All right, well, Well, I know it's been a very packed day and a half, and so I understand everyone has had a lot to think about over the past 2 days. Are there any closing comments here on the panel that you'd like to make? I think you've made quite a few, but if there's anything else that came up.
I just wanted to say that, you don't have to have a qualified method to use it in an application. So I just, people can use any method that they think are, going to help them demonstrate the safety and efficacy even of their product, and it doesn't have to have formal qualification. So I don't want people to think that there's a limitation that FDA requires you to have gone through a formal validation process in order to use a method. As long as you can have some internal validation which demonstrates the performance of your model and the kind of, the value of it, I don't think that there is any barrier to that being used in any application.
Definitely, from the 3 different routes for a DDT acceptance that Jeff brought up yesterday, definitely.
I don't like sitting in silence, so I have to say one last thing. It's a big thank you to the CPATH group, actually. I think, it goes without saying that you've put a lot of thought into this. It's been, a process that started many, many months ago. I've participated in many of the calls online, and I think coming here, seeing all the stakeholders together and listening to the dialogue's been really helpful, and I said to Nick, he knows, but I was nearly ready to pack up and go home yesterday. I thought, oh my God, we're never going to get anywhere. And actually he said, stay with it. Your flight's not until tomorrow night, which it is. It's delayed actually. So hope you didn't, hope you didn't jinx it. But to go through the full circle, I feel much more positive. So thank you very much indeed.
Thank you, Lorna.
And perhaps another closing thought is that this was only the beginning, and that we're going to eventually fill the need to have the framework, but the framework itself is going to be its own evolving entity. And we took the first step. So kudos to you guys for sticking it out for these couple of days. Liver was the first case study. It'll be teaching a lot of other organ toxicities and tissue toxicities, a lot of things, but we had to take this step. So there's going to be more to come.
Thanks. And with that, if the panel would like to go ahead and step down, we can close today's meeting.
We did just get a question from the remote attendees. No, sorry. Okay. Question on the last comment about pharma not using a method until it's qualified. Can you clarify that the use of a new method must still be accommodated by animal studies in the preclinical trial stage?
Yes. I mean, I think that what I was speaking to, at least, is if you have a tool that you propose can address a component of your IND package, and it's something that hasn't been qualified, but you've built up a level of evidence and competence in that model, it's something to bring forward as a conversation in a pre-submission to the FDA to open up that conversation on an individual case-by-case use. I hope that answers the question. I'm not sure if anyone else from FDA. Not sure which reviewers we have left. All right. Well, I wanted to open up my closing by again thanking everyone in this room for making the trip here and meeting in person. These workshops, I know that we've become quite accustomed to meeting virtually, but sometimes I think, especially at an early stage like this, we really need to come together and have multiple groups going through the issues that they see, the positives that they see, the negatives that they see, and where we can find compromise and appropriate paths forward. You know, obviously we didn't answer all of those questions today. This was one step in the path. And so thank you to all of you for contributing and participating in today's meeting. Over the next few months, we will be working to refine this framework, and I will be looking to everyone in this audience to participate. We will be continuing the regular recurring meetings that we have. I think we'll probably be refining them down to 3 different areas now. It'll be around appropriate contexts of use, analytical considerations, as well as biological performance considerations, and incorporate in as well the wonderful handout that Tina provided us around building a framework and addressing an unmet need. So that will be the work we have in front of us over the next 6 months. And so I would also like to thank the fact that we have several clinicians able to join us. Eric, thank you for sitting through to the end with us. I think, you know, in this area, we are frequently working with a lot of people who are in investigative toxicology and early discovery, and having an additional perspective from someone who's working in phase 1, phase 2, phase 3 clinical trials to inform on where they see unmet needs and applications for complex in vitro models is crucial to advancing the field right now. So thank you, Eric, thank you, Mark, and thank you, Skip, for joining us. And with that, have a great weekend, and I wish you easy travels back to your homes.